How's it going, class? So this is going to be a bonus lecture looking at muscle contraction. I realized that going through the muscle contraction and the muscles move us lecture wasn't super in depth, and it doesn't really give you a lot of detail in what's really occurring, as well as we didn't really talk about what ATP is doing and how it's involved in muscle contraction. So this is going to be kind of a short bonus lecture going through the different steps, explaining it in more detail so that you guys can understand it. Because I think this is a very fundamental process in how we move. So obviously, when you're moving during exercise, it's because your muscles are creating force, they're producing force. And this is going to explain to you how that impulse essentially gets from the brain down the central nervous system, spinal cord, through the alpha motor neuron, and then into the muscle and the different moving parts that allow it to go through its contraction and produce force for you, as well as if you are listening to this bonus lecture, I'm hoping that you're one of the more involved students. And I'm going to give you this hint right now. On the exam, there's going to be 43 questions, something like that. But the basis of what I'm trying to say here is that there is going to be 3 essay questions on the exam. Now, I'm going to post probably 3 or 4 bonus lectures throughout the first test session here. And there's going to be 3 essay questions on the exam. Now, I don't think I need to tell you what 2 plus 2 means, but I would assume from what I'm saying that you're understanding that these bonus lectures are probably pretty important for your exam. So without further ado here, I'm going to jump into the bonus lecture here. All right, so muscle contraction, right? Bonus detail lecture. So what we're going to be talking about here is just what is occurring, right? And so basically we're going to start here again, the structure of the muscle. So the muscle fascicle, muscle fiber, myofibril, myofilaments, and then sarcomere. If you want to think about this, you can think about this in reverse fashion as well, right? You can think about it like the force is being produced here in the sarcomere. Oh, I gotta respond. Trying to draw, I'm just clicking through. Is being produced here in the sarcomere, okay? And the force itself, right? The sarcomeres are all within the myofilaments. So the sarcomeres are what are actually going through the contraction, right? Which is causing the myofilaments to shorten, which is causing the myofibril to shorten, the whole muscle fiber shorten, the muscle or the fascicle, and then the muscle itself, right? So it's being broken down into its individual parts here, but these sarcomeres are prevalent in all of these parts, right? You should be able to see this broken down smaller and smaller, and then follow it back from the smallest point to its largest point and say, okay, hey, this muscle here is comprised of millions of these overlapping and repeating sarcomeres. So the sarcomere is the smallest contractile unit of the muscle. This is where all the force you use for all movement is generated. This is what we're going to be focusing on for this, and this is going to be at least the end point of what we're talking about. We're going to talk about how we get to this, and then what occurs here to generate that force, which is allows your muscles to shorten and move, which is allows you to move around your environment, lift weights, play sports, do whatever it may be. So the action potential is what we're going to start with, right? And the action potential begins in the brain. Your brain is where you think of what you want to do, where you want to do it, how you want to do it. So the CNS action potential begins in the brain. It is then going to come down your spinal cord here, right? So it's going to come down the spinal cord here until it hits the level of the spinal cord where it wants to be transferred to a muscle. So the different vertebrae in your spinal cord have foramen on the side of them that allow different nerve roots to escape. So we see these nerve roots right here, so be the anterior and the posterior nerve roots. This is these escape out through your vertebra, right? Your vertebrae. So depending on what level, let's say it's the arms at like a T6 level or the legs at like the L3 or L4 level, it's going to be the same process here. It's going to come down the spinal cord and then it's going to exit the spinal cord at whatever level you need it to. For the purposes of this lab, we're not really going to talk too much about the dorsal root ganglion. This is mostly sensory inputs. But we're going to be talking about the motor output, the motor neuron, the alpha motor neuron comes down the spinal cord, synapses onto the alpha motor neuron, and then the alpha motor neuron is going to travel to the muscle that we want to be activating. So we don't really need to know, I'm not going to talk about what's occurring right here. You should know action potentials from any physiology classes you've taken before. We're not going to talk about the synapse within the central nervous system, but you should know that the alpha motor neuron is depolarizing and it is sending a signal to the muscle here, and it is releasing what we'll talk about in a second here. So when it gets to that point, right here, you should see, right, this motor neuron is now depolarized and it is releasing these little vesicles, these little bubbles of acetylcholine, okay? And this acetylcholine is then transferring, or it's flowing across the synaptic cleft or the synapse here. And so you need to release enough acetylcholine so that when it flows across the synapse, it can bind to these acetylcholine receptors and cause a depolarization of the muscle membrane itself. So when enough acetylcholine is released, or the stimulus from the outer alpha motor neuron is strong enough, okay, it's going to release this acetylcholine into the synapse. And if enough of this acetylcholine is released into the synapse, it's going to bind to the muscle fiber here, and it's going to cause a depolarization of the plasma lemma, of the muscle fiber or the muscle cell itself, okay? And so when that muscle cell then becomes depolarized, and it depolarizes in the same way as a neuron in all or none potential, you either have enough stimulus for it to depolarize entirely, or it's not going to depolarize at all. When the muscle fiber itself is depolarized, you're going to get a calcium release from the sarcoplasmic reticulum, okay? So we see here, I can get my stuff out of the way here so you can see what you're doing, right? We see here, right, we have our sarcoplasmic reticulum here in the yellow, and then up here, it's in this light blue, right? We see this is wound and interwoven around all these muscle fibers, around all these myofibrils, okay? So this sarcoplasmic reticulum releases calcium, and that calcium is what travels down the T tubules. That's what travels down these T tubules and moves throughout the muscle cell or muscle fiber, okay? And you can see here as well, these transverse tubules, these T tubules, this yellow as it is here, it transfers and is like a spider web network through the muscles. It's like a highway that allows this calcium to get wherever we need it. And then the calcium, right, is released into the sarcoplasmic reticulum. And where does it go next? Well, we should know, if we've been paying attention to that first lecture, is that the calcium is what's required to move the troponin and tropomyosin off of the actin-myosin binding sites. So we see here, we should see in this top little diagram here, as well as we can look at this picture here, is that these free-floating calcium ions bind to the troponin, which are going to move the tropomyosin off of the actin-myosin binding sites and allow for the myosin heads to attach to the actin, okay? So the myosin heads cannot attach to the actin until calcium has bound to the troponin, which moved the tropomyosin and allowed for the attachment. And if there's no attachment, that means there's no force production, okay? So if those myosin heads are not allowed to attach to the actin because the calcium is not bound to the troponin moving the tropomyosin, that means that we get absolutely no force production. So this calcium release is extremely important for our force production, as well as when we talk about in fatigue, right? We'll talk about how hydrogen ions and different things interfere with this calcium affinity for the troponin, which is going to cause or manifest some of the feelings of fatigue. And so we said calcium is bonding. That allows the myosin to attach, right? And then we see the myosin go through a power stroke, which is what moves the actin, which is what shortens the filaments, which is what essentially force production is. And then, as I said in the first lecture, people understand that the muscle is shortening, but they don't really understand how. Most people think like the muscle is kind of crinkling up like a ball of paper. What really is happening is that these filaments are just sliding past one another. And because this myosin, right, this myosin filament here, this heavy or thick filament, is anchored in the middle, and these actin filaments can move and become closer together, that's what the shortening is. You essentially have an anchoring point right in the middle, and then the actin filaments shorten towards that anchoring point. And if this is repeated in parallel hundreds, thousands, millions of times, you get massive shortening of your muscles. So talking about it a little bit further in detail here, how do we get this power stroke to occur? This is something that was really missed in the first lecture, and something that I really want to cover a little bit more detail because it makes more sense once you understand it from this point of view. So we'll talk about it in lectures to come, as well as you should probably know that ATP is the energy currency of the body. It's how you get things done. Okay? And in the first lecture that I sent out about this, I didn't really talk specifically about how ATP is involved in this and what it does and what's occurring. And you're not going to need to know the specific enzymes here. You're not going to need to know that ATPase is involved and know that there's different ATPases, right? That's good and pertinent information for you. But we're going to talk about just the basis of what's going on here. So we're going to start in this non-attached hydrolyzed state here. So in this state here, right, ATP has already been hydrolyzed into ADP and inorganic phosphate, or PI as it's written. So this is the state in which we're going to start. Usually, realistically, what's occurring is you're kind of starting at stage 4 here. This is a good way where a lot of diagrams start. However, it doesn't really matter. A lot of places like to start at 1 here, just because you're thinking about, okay, when does the cross-bridge stroke initiate? How does it happen? Then how does it recover? So really, we're going to start from point 1 here. ATP has already been hydrolyzed into ADP and inorganic phosphate. Now, the hydrolyzation of that ATP into ADP and inorganic phosphate is what changes the affinity of the myosin and binds it to the actin. But again, remember, this myosin cannot bind to this actin unless this calcium had already bound to the troponin. So if there's no calcium, even though these ATPs are getting hydrolyzed, they're just sitting there and waiting like you see in stage 1. And that's why generally these models begin at this stage 1 right here, is because usually ATP is hydrolyzed and ready to go. We just need to wait for the calcium to be released from the sarcoplasmic reticulum and bind to the troponin for the muscle contraction to incur, right? So the ADP, inorganic phosphate, changes the affinity of the myosin head. The tropomyosin has been moved, which means it now has attached, okay? So now it's attached. It's in this cocked and ready position. It's attached to the actin, and it's waiting, right? Now, the ADP and inorganic phosphate get cleaved off of the myosin head, which causes it to go through a conformational change and change its shape. So usually, if you remember biology, how you change the shape of proteins is by binding things to them or removing things from them. It's the same thing here, right? The myosin protein is in this shape. It's ready, cocked, ready to go. When the ADP, inorganic phosphate, are cleaved off, you get a power stroke. You get basically a flick of a finger, right? Except in the opposite direction, more like the bend of a finger, okay? So you get the bend of a finger, which is what pulls the actin along, right? That's essentially what's happening. The bend of the finger pulls the actin along. What happens next is you need ATP to reattach to the myosin head. So that's not really shown here, but really what happens is this stays locked here. This stays locked on here until this ATP binds to it. So the ATP binds to it, right? Binds to it. It releases from the actin. So if you're looking at me in the picture here, it releases from the actin and also causes it to go back to a Cox position, right? So we just went through the contraction. We're waiting right here. ATP binds, it causes it to release, and also to go back to the Cox position, right? And then from that Cox position, we're sitting there with a myosin head and ATP on it. That ATP is hydrolyzed into ADP and inorganic phosphate here. So as you notice, 5 and 1, right? 5 and 1 are basically the same step. It's just that 1 is specifying that calcium bound, and 5 is specifying that we're recovering this process. And so as long as calcium stays bound, it's not like one calcium needs to bind for every ATP. As long as calcium is still being released from the sarcoplasmic reticulum, as long as it is still bound to troponin, the tropomyosin is still moved, this power stroke or this cross-bridge cycling is going to occur over and over and over again. The limiting step here is when that calcium is removed. When the calcium is gone, the tropomyosin and troponin move back, and now the cross-bridge cycling can't occur. But because there's still readily calcium available, this power stroke continues over and over, right? We see continuing pulling of the myosin and actin, right? And this is happening in sequence and in parallel all over the muscle fibers. You really have millions and millions of these little fingers just pulling on the actin, which is shortening the filament, which is allowing you to produce force and go through general movements. And so with this too, if any of you know what rigor mortis is, I'm sure this is a fun fact that's told in all of your classes, but any know what rigor mortis is after a living individual, usually a mammal, dies, right? They go through a state of rigor where they're locked in a muscle position. That is because, right? That is because the ATP is no longer binding to the myosin heads, allowing for the muscle to detach, allowing for the myosin to detach from the actin. And so essentially you're stuck in that conformational shape where the myosin is attached to the actin. And that essentially is what is preventing those muscles from moving. So if you're a hunter, hopefully none of you have ever experienced an actual dead body, but if you're a hunter and you've experienced dead bodies like an animal, deer, birds, anything like that, you understand that they get very stiff afterwards. Or if you watch crime shows, if you're a CSI person, which I'm sure is where most of you have heard this, you understand what rigor mortis is, right? And that's essentially what's occurring here, right? Or essentially what's occurring here is just the muscles themselves are basically stuck together. The muscle proteins are stuck together. And so, right, to reiterate this, we're talking about ADP, right, in an inorganic phosphate, they're attached to the myosin head. Myosin head then attaches to the actin. When the ADP and inorganic phosphate are cleaved off, we get the power stroke. When ATP bonds to that myosin head, it moves off of the actin, goes back to its cock-ready position. ADP, inorganic phosphate, or excuse me, go back to the cock-ready position where it's hydrolyzed, and then ADP and inorganic phosphate make the myosin head more affinity for the actin, which allows it to bind. And then again, this process occurs over and over and over until there's no longer calcium available. And why would calcium no longer be available? Well, we can think about this in an opposite manner of what we did before as well. Okay, so you can think about this. What happens? So the CNS sends the active potential, the alpha motor neuron, all the way down. That's how we get a myosin head power stroke and shortening. When this signal stops, the alpha motor neuron is no longer stimulated by the central nervous system, right? By the primary motor neuron, the first order motor neuron, right? When the alpha motor neuron is no longer stimulated, acetylcholine is no longer released. When acetylcholine is no longer released, the sarcolemma, right, the plasmolemma, the sarcoplasmic reticulum of the muscle cell are no longer going to release calcium ions. When the calcium ions are no longer being released, they can't bind to the troponin, which opens up the myosin-actin binding sites. If the troponin is not moved off the myosin-actin binding sites, myosin heads cannot attach to the actin, and that means that the myosin power stroke cannot occur. So this is basically just a step-by-step process that begins from the CNS and then is altered by cessation of signal from the CNS. So you begin muscle contraction by the brain saying, all right, let's do some contractions, let's move. And then you stop movement by the brain stopping sending that signal. It doesn't actually send it really a signal that tells you to stop this process. It just stops sending the original signal that we sent out in the first place. If that makes sense. So that, in essence, is the basis of what we wanted to talk about for this lecture. I want to talk about this a little bit more in detail. There is a lot of links out there on YouTube that can show this process in real time, as well as explain it in a very similar way to myself. So if I'm not explaining it in quite the way that you think, if you're not explaining the way that you like it, there's other references out there. There's other sources out there for you to do this, as well as they're going to show you videos. And the best way to probably do this is to listen to my lecture, understand on a surface level what I'm saying, get this first interpretation of what the information is, and then go and look at other YouTube videos or go and look at other videos within the course and say, okay, now what's occurring when I see this actually in motion? Explaining muscle contraction as a concept in basic 2D and through verbal language is tough. You kind of have to show what's occurring, show how the filaments are sliding past each other. That's why I use the finger and the arm movement, right? Very, very simple, breakdownable, digestible ways of trying to understand this complex mechanism, right? So that's going to be the end of this first little bonus lecture. If you guys have additional questions about this, like I said, I'm going to say this a million times, I love talking about this stuff. I love working with you guys. If you guys want to meet in person, if you want to meet over Zoom, ask me questions, talk about this, ask how it relates to everyday living, ask how it relates to your sport, whatever it may be, please reach out to me and I will do my best to help you out. As well as after this first week here, if you guys still have any questions about how the course functions and how things are activated and done, please reach out to me as well. And then finally, if you have not yet signed up for the perusal or you're having troubles, please make sure that you are logged out of any account that you made, and then click on one of the links that I sent you. So click on the link that I sent in the announcements or email, or click on the link for the introductory video, and then sign up and see if it'll take you there. If you're still having troubles, email me, and I might have you reach out to perusal support. Otherwise, look forward to continuing to work with you guys this semester, and I'll see you in the next lecture.